Good evening, all of you. Am I audible? If I'm audible, can I get a confirmation in the chat box, please? Can I see a thumbs up? Am I audible? Okay. So, if you're joining the session for the first time, thank you, Anisha. So, if you're joining the session for the first time, this is Dr. Shanmugapriya. I'm professor and head of the Department of Biochemistry. Good evening, Anisha. Good evening, Bablu. I'm professor and head of the Department of Biochemistry at Government Thoothukudi Medical College, and I'm your Unacademy educator for Biochemistry. And today, the session, I planned for a 1-hour session, and this session will be all about genetics. So I plan to start from the very basic fact of genetics, wherein I will be telling about nitrogenous bases, nucleosides, nucleotides. Then I will tell you how a polynucleotide chain is formed, then DNA. We will know a bit about replication, and then I will be telling you about application aspects of genetics. Okay, so I'll be telling about properties of genetic code, self-splicing, introns. So all high-yield MCQs will be discussed today. So that's the plan for today. Good evening, Dayananda. Yeah. So that's the plan for today's session. Before we start the session, let me tell you a few facts quickly about Unacademy plans. So a reminder for all of you to participate in NEET PG 2022 All India Mock Test, which is on Feb 26th. And the time is 9 a.m. And if you're asking me why should we attend this test, for two reasons. One is the analytics will clearly tell you, it will give you valuable insights into your preparation. That's point number one. And more important than that is you will be provided a video solution for every MCQ. So you don't have to hunt for the reason or for the explanation for any of the questions. A videographed solution will be provided for every MCQ. Okay, so that's about All India Mock Test. But apart from that, there are multiple tests which are happening on the platform. There are subject-wise tests, grant tests. So I recommend that you attend as many tests as possible. And if you're a NEET PG aspirant, there are multiple batches which are happening on Unacademy platform. I would say the gold standard would be all educator revision batch. And what is so special about this batch? Because in this batch, there are just 4 hours of live high yield MCQ session. Yeah, every day there are only 4 hours of live session, which means the remaining time you can have for yourself for self-study. Okay, and the schedule will be exactly fitting into the expected weightage that is provided for every subject in NEET PG papers. And after every subject gets over, there is also a subject-wise test that is conducted. And after the subject-wise test, an explanation will be provided for every MCQ that is asked in the test. And apart from this, you will be able to attend 8 grant tests. So by the end, you will be very confident of appearing for your NEET PG 2022 exam. Okay. So this is about the all educator revision batch. And apart from this, we have multiple batches as I told you. There is a special image based question batch. There is a high yield MCQ marathon batch. Previous year questions batch, all for NEET PG aspirants. And if you're an FMG aspirant, there are 2 batches. One is a comprehensive batch, duration would be 4 months, and the other one is duration would be 10 months. That's for the next FMG. Okay. And there is also a next subject-wise batch, that is the duration of the course would be 1 year. And to be able to participate in any of these batches, you should have either a plus subscription or an iconic subscription. Iconic subscription gives you an access to not only Unacademy platform, it also gives you an access to PrepLadder platform. And there is a current offer that is going on, limited time offer that is going on for this plus subscription. It is valid till 23rd of February, wherein if you buy 3 months subscription, you get additionally 1 month subscription free. 12 month subscription, you get 2 months subscription free, and so on. And on top of this, if you use any of the educators code, you get 10% off. You can use any of my codes. One code is my name C Shanmugapriya, the other one is Biochem Live. You can use any of those 2. Rapid revision, yes, I've done rapid revision. I think I did it 7 days back. Yeah, entire biochemistry in 90 minutes was my plan, but it got extended to I think 120 minutes. So it was done as 2 sessions. You can check those videos, Dr. Abhishek. And after this session, I will start doing mixed bag MCQs. Probably that will also help you. Okay, on YouTube. So that's all about Unacademy plants. Let's start discussing MCQs on genetics now. Okay, so listen to this carefully. All of the following are purine bases except. Yeah, I will wait for 1 minute for you all to answer. All of the following are purine bases except. A very basic question. Yeah, what is answer? The answer is uracil. Why is it uracil? Because it comes under cut. How do you remember pyrimidine bases? Pyrimidine bases can be remembered using a mnemonic. What is a mnemonic? Cut. What does cut stand for? Cytosine, uracil, thymine. Very good, all of you. Excellent. Sushma, Bharat, Aruna Devi. Yes. Prithvi, Dancer, Anushka. Okay. So, what are the pyrimidine bases? It is cytosine, uracil and thymine. Okay. And let me tell you few facts about nitrogenous bases. What are the two types of nitrogenous bases? Purines and pyrimidines. What is the difference between a purine and a pyrimidine? I would like to tell you all that only when you have these basics strong, you will be able to apply it. Yeah. So bear with me for another 5 minutes until I finish telling you about these basics. Then we'll start applying it. Okay. So the two purine bases, the two types of nitrogenous bases are purines and pyrimidines. Purines with a smaller name have a bigger structure. They have 2 rings and 9 atoms. Yeah, purines have 2 rings and 9 atoms. Pyrimidines with a longer name have a smaller structure. They have single ring and 6 atoms. Can you see that here, all of you? Yeah, purine, a smaller name, has got a bigger structure, 2 rings and 9 atoms. Pyrimidine, a longer name, has got a smaller structure, single ring and 6 atoms. Now, what are the pyrimidine bases? I just now told you the mnemonic that you can have for pyrimidine bases is cut. So, what are the cut bases? Cytosine, uracil and thymine. Now, about purine bases, listen to this. There are 2 types of purine bases. Yeah, generally we think there are only 2 purine bases, adenine and guanine, right? Whenever I ask you what are the purine bases, students generally answer it as adenine and guanine. But I want you to know that there are 2 types of purine bases. One is major purine bases, the other one is minor purine bases. So what are the major purine bases? This will include the adenine and guanine, which every one of us know. Okay. Now what are the minor purine bases? Minor purine bases include hypoxanthine, xanthine and uric acid. So tell me, what are the minor purine bases, all of you? Hypoxanthine, xanthine and uric acid. Now, what is the significance of major purine bases, adenine and guanine? These are present in any polynucleotide chain, be it a DNA or RNA. All these polynucleotide chains will be made up of adenines and guanines. Now, what is the significance of the first minor purine base, hypoxanthine? Listen to this. This has been asked before. The significance of the minor purine base hypoxanthine is that its nucleoside is inosine. Do you all understand this? Its nucleoside is inosine and its nucleotide will be called as inosine monophosphate, about which I will tell you in the last MCQ for today's session. Okay, so for now, what will you remember? I want you to remember that the significance of hypoxanthine is its nucleoside is inosine and its nucleotide is inosine monophosphate, which carries few significances. Okay, good. Now, what is the significance of the second minor purine base, xanthine? Have you heard of Kefin? Yeah, Kefin, theophylline, theobromine. Kefin found in coffee, theophylline the bronchodilator. Yeah, theophylline, theobromine found in tea. All these 3 are derivatives of xanthine. Kefin is 1,3,7-trimethyl xanthine. When all 3 get methylated, you don't have to write anything. I will be providing that in the PDF. Listen to this now. What is Kefin? Kefin is 1,3,7-trimethyl xanthine. Theophylline is first 2. 1,3-dimethyl xanthine is theophylline. 3,7-dimethyl xanthine is theobromine. Is that here? So memorize it. Kefin, theophylline, theobromine are all derivatives of xanthine. Okay. And the third minor purine base, uric acid, significance is very simple. All other purines on catabolism will give rise to uric acid. Okay, so that's all about purine bases. So have you understood this clearly? Yeah, what are the 2 types of nitrogenous bases? Purines and pyrimidines. Pyrimidines will include cut. What does cut mean? Cytosine, uracil and thymine. Purine bases, we have major purine bases and minor purine bases. Major purine bases include adenine and guanine. Minor purine bases include hypoxanthine, xanthine and uric acid. If you ask what is inosine, what will be your answer, all of you? If I ask you what is inosine, what will be your answer? Inosine is a nucleoside of hypoxanthine. Okay. It's a nucleoside of hypoxanthine. So what is answer? The answer is uracil. Now what are the other points to remember? What are the other possible questions that can be asked as far as nucleotide chemistry is concerned? So can you all tell me, how do you convert nitrogenous bases into nucleosides? Can anyone tell me, how do we convert nitrogenous bases into nucleosides? Whenever these bases get attached to a sugar, right? Whenever bases get attached to a sugar, you will call it as a nucleoside. Now what is a sugar that gets attached? Yeah, what is a sugar that gets attached? The sugar is either ribose or deoxyribose sugar. So can you tell me what is deoxyribose sugar? Ribose is a pentose. It has got 5 carbon atoms and all these carbon atoms will have hydroxyl groups. When you call something as deoxyribose, it means the second carbon atom alone has lost its oxygen. Yeah, if it's deoxyribose, what does it mean? It means the second carbon has lost its oxygen. So whenever I call something as deoxyribose, I mean to say it is 2-deoxyribose. Very good, Bharat. It is 2-deoxyribose. Now what, it is not in the 5th carbon, Ibiza. 5th carbon is phosphate. I will tell you about that later. Okay. So deoxyribose. So tell me, what is the linkage that is formed here? When base gets attached to a ribose or a deoxyribose sugar, what is the linkage that is getting formed here? This has been asked twice in NEET. Yeah, the linkage is beta N glycosidic linkage. All of you tell me, what is the linkage that is formed here? Beta N glycosidic linkage. I don't have time to explain you why it is called so. For now, to remember, we know there should be a sugar. If there should be a sugar, won't you call it as a glycosidic linkage? Yeah. So what is the linkage that links the base with the sugar? Base with the sugar is linked by beta N glycosidic linkage. Okay. Very good. Now from nucleoside, how will you convert it into a nucleotide? From a nucleoside, how will you convert it into a nucleotide? By attaching a phosphate group. So from where do you think the phosphate group gets attached? All of you memorize this, or just remember, ribose 5-phosphate is like a foundation. Yeah, ribose 5-phosphate is like a foundation based on which you build all nucleotides. So it is ribose 5-phosphate. So tell me, where will the phosphate group get attached? All of you, very good, Prithvi Sai. Where will the phosphate group get attached? The phosphate group gets attached to the 5' hydroxyl group of nucleoside. I hope now Ibiza has understood it. It gets attached to 5' hydroxyl group of a nucleoside for you to get a nucleotide. And the linkage is called as phosphoester linkage. Why do you call it as a phosphoester linkage? Because a phosphate group is an acid group. Acid group is getting attached to an alcohol. Acid plus alcohol is an ester linkage. So tell me, what is the linkage that is present in a monophosphate nucleotide? Good evening, Vairam. Yeah, what is the linkage that is present in a monophosphate nucleotide? It is a phosphoester linkage. And I want you to remember that it is a monophosphate nucleotide that is present in all polynucleotide chains. Do you all understand this? Suppose I say sequence of a particular polynucleotide chain is ATGC, AT, TA. I'm just telling you that this is the sequence of a polynucleotide chain. What does it mean? It means it has got AMP, TMP, GMP, CMP and so on. So tell me, what is the form of nucleotide that is present in any polynucleotide chain? It is a monophosphate nucleotide. So now I am going to tell you how do these monophosphate nucleotides get linked to form a polynucleotide chain. Okay. For understanding this, you will have to know that for any substance to get linked in a poly form, the group it would prefer to have is a free hydroxyl group. Do you all understand this? For any substance to get linked in a poly form, the group it would prefer to have is a free hydroxyl group. And can you see that in this monophosphate nucleotide, the only hydroxyl group that is free is the 3' hydroxyl group? Yes or no? What has happened to the first hydroxyl group? The first hydroxyl group of the sugar is involved in a linkage formation with the base. So can you tell me what is the linkage here? The linkage between the sugar and the base. What is the name I told you? The name is beta N glycosidic linkage. Okay, it is beta N glycosidic linkage. And can you rely on the second hydroxyl group, all of you? Can you rely on the second hydroxyl group? No, because if it's a deoxyribose, I said second carbon will not have OH, it will just have a H. Okay, can you rely on the last hydroxyl group? No. Why can't you rely on the last hydroxyl group? Because it is involved in phosphoester linkage formation with the phosphate group. So what is the only hydroxyl group that is always free? Yeah, the only hydroxyl group that is always free is 3' hydroxyl group. This 3' hydroxyl group will get involved in linkage formation with 5' phosphate group of the next nucleotide. Can you see that here? Yeah, the 3' hydroxyl group of the first nucleotide is involved in a linkage formation with the 5' phosphate group of the next nucleotide. So can you tell me what is the linkage that is getting formed here? Alcohol plus acid is an ester linkage. Don't you know that there is already another ester linkage on the other side? Yeah, there is another ester linkage on the other side. So this is a second ester linkage that is formed. So can I call it as diester linkage? Yeah, there is already another ester linkage. I am forming a second ester linkage. So what will I call it as? I will call it as phosphodiester linkage. And what is the numbering, all of you? Very good, Bharat. What is the numbering? It starts from 3' carbon atom, ends in 5' carbon atom. So the answer is 3', 5' phosphodiester linkage. So have you all memorized it? Yeah, have you all memorized it? We generally underestimate such facts. Only because we underestimate such facts, we are not able to understand concepts. So please memorize it. What is the linkage that is present between individual nucleotides in a polynucleotide chain? It is 3', 5' phosphodiester linkages. Okay. And can you see that here? This is a polynucleotide chain. This polynucleotide chain has got AMP, GMP, CMP and TMP. And if you take the first nucleotide, what has happened in the first nucleotide? There is a 3', 5' phosphodiester linkage formation. And because you have formed a 3', 5' phosphodiester linkage, which group is free, all of you? All of you, which group is free? The 5' phosphate group is free. And that end where the 5' phosphate group is free is called as the 5' end. Do you all understand this? What is a 5' end in a polynucleotide chain? It is the end where the 5' phosphate group is not involved in any linkage formation. And if you take the last nucleotide, in the last nucleotide, which group is free, all of you? The 3' hydroxyl group is free. This is called as the 3' end. So can you see that any polynucleotide chain synthesis, listen to this carefully, this is why I am telling you all these facts. Can you see that any polynucleotide chain synthesis should begin from 5' end and it should end in 3' end? Yes or no? It should begin in which end? It should begin in 5' end, it should end in 3' end. And that is why we always say all polymerases. What are the polymerases you know of? DNA polymerases and RNA polymerases. So all these polymerases synthesize a new strand only in which direction? They all can synthesize a new strand only in 5', 3' direction. Is that clear to all of you? Yeah, please understand this. All polymerases synthesize a polynucleotide chain only in 5', 3' direction. Okay. So that is all about a polynucleotide chain. Okay. Have you understood it? Yeah. So if this is a polynucleotide chain, then how will you get a double-stranded DNA? This is one strand. Yeah, what I showed you here is one strand. So how will you get a double-stranded DNA? When one more strand gets attached to this strand in an anti-parallel fashion. What is anti-parallel fashion? I showed you this as 5 prime 3 prime. So the other strand will be 3 prime 5 prime. And both these strands will be linked by what linkages? Both these strands will be linked by hydrogen bonds. This is how you get a double-stranded DNA. Yeah, this is how you get a double-stranded DNA. So can you quickly summarize all that I've told you so far? Yeah, so nitrogenous bases will get attached to ribose or deoxyribose sugar to form a nucleoside, right? And the linkage is glycosidic linkage. What linkage is it? It is beta N glycosidic linkage. And from nucleoside, how do you form a nucleotide? By attaching a phosphate group. Where will you attach the phosphate group? You attach the phosphate group to 5 prime hydroxyl group. And what is the linkage that you get here? That is called as, don't call it as diester linkage, that is a phosphoester linkage in a monophosphate nucleotide. And what is the form of nucleotide that is present in any polynucleotide chain? Every polynucleotide chain will have only monophosphate nucleotides. Tell me, what is the linkage that is present in a monophosphate nucleotide? Yeah, what is the linkage that is present in any polynucleotide chain? The numbering is very important. It is 3 prime 5 prime phosphodiester linkages. So any polymerase, it can be a DNA polymerase, it can be RNA polymerase, any subtype it is. If they ask you what is the direction in which your polymerase can synthesize a new strand, close your eyes and say it has to start from a 5 prime end, it has to end in 3 prime end. So it is 5 prime 3 prime polymerase activity. Okay, when two such strands get associated with hydrogen bonds in between, we get a double stranded DNA. Okay. Now what is chromosome? Can anybody tell me what is chromosome? Anybody who is listening to this session, what is a chromosome? The chromosome which you draw with a centromere, a short arm and a long arm is nothing but a long double stranded DNA. Do you all understand this? The chromosome which you draw with a short arm and a long arm is nothing but a long double stranded DNA which has got condensed with the help of proteins. Do you understand this? Yeah. So what is a chromosome? Every chromosome is a double-stranded DNA which has got condensed with the help of proteins. Okay. So that's about a basic introduction to genetics. Okay. Now this is the next question. Try answering this, all of you. The histone that is not present in a nucleosome is? The histone that is not present in a nucleosome is? Can anybody try answering this? Very good, Bharat. It is H1 histone. Yeah, histone that is not present in a nucleosome is H1 histone. So this is based on how you form a chromosome. So just now I told you chromosome is nothing but a long double-stranded DNA which is condensed with the help of proteins. Now why should you condense the DNA? It is because if you take any chromosome, chromosome 1 or chromosome 2 or chromosome 3, if you uncondense the chromosome, that is going to measure a few meters long. Now can you pack such a long chromosome inside a nucleus of few microns in diameter? You cannot do that. And that is why we are trying to condense the long double-stranded DNA to form a chromosome. So how do we condense the long double-stranded DNA? That is done with the help of histones. So what are histones? When I say histones, they are basic proteins. Yeah, what are histones? Histones are basic proteins in the sense they are all positively charged. Now how about the charges of DNA? Can you all answer this? Histones are positively charged. How about the charges of DNA? Because DNAs have phosphate groups, so there is going to be an attraction. Yeah, there is going to be an electrostatic attraction between positively charged histones on one hand and negatively charged DNA on the other hand. So that a segment of double-stranded DNA, because of the electrostatic attraction, a segment of double-stranded DNA gets wound around histone. So this gives the first level of compactness to your chromosome. Do you understand this? This gives the first level of compactness to your chromosome. So can you tell me how many types of histones do you have? Is there a lag in the stream? One second. Is there a lag in the streaming? So am I seen now? Can you all see me? Okay. So I was telling you about how chromosome is formed, right? That is where we stopped. So I was telling you about histones, and I said there are 5 types of histones. Yeah, let's continue with this now. So how many types of histones do we have? There are 5 types of histones. Yeah, what are the 5 types of histones? We have H1, H2A, H2B, H3 and H4. Yeah, how many types of histones do we have? H1, H2A, H2B, H3 and H4. Out of these 5 types, all except H1, the remaining 4 form dimers. When 4 form dimers, what will you call it as? You will call it as an octamer. So this histone octamer sits in the center, rich in positive charges. And this positive charge, all these positive charges will attract a segment of double-stranded DNA which gets wound around. Okay, so this string on bead appearance, what is the string here? The string here is a double-stranded DNA. What is the bead here? The bead here is histone octamer. So this string on bead appearance is called as a nucleosome. So that is where you get the first level of compactness. So can you tell me which histone is not present in a nucleosome? It is H1 histone. And I want you to visualize that you have taken a long thread. Yeah, just visualize that you have taken a long thread. On this long thread, you have placed beads at various places and you have allowed the thread to get wound around those beads. Analogous to that, what we have done here is we have taken a long double-stranded DNA. On this long double-stranded DNA, we have placed histone octamers at various places and we have allowed the double-stranded DNA to get wound around histone octamers. So several such nucleosomes exist. Do you all understand this? Several such nucleosomes exist and that will be connected by a linker fragment. Yeah, all these nucleosomes will be connected by a linker fragment. And H1 histone is present only in the linker fragment. So tell me, if H1 is not present in nucleosome, where do you find H1? You find H1 in the linker fragment. Now one such fibril is called as a 10 nanometer fibril because the width is just 10 nanometer. You call it as 10 nanometer fibril. Now a segment of 10 nanometer fibril gets folded on itself like this. Yeah. A segment of 10 nanometer fibril gets folded on itself. Now the width increases. This is why you get the second level of compactness. Don't you think this looks like a beta pleated sheet? Yeah, a segment of 10 nanometer fibril getting folded on itself, giving you the second level of compactness. This is called as 30 nanometer fibril. Now you take this 30 nanometer fibril and you form loops on chromosome scaffold. In your nucleus, you have 46 chromosome scaffolds, one for every chromosome. What is a scaffold? It's a tubular protein. So this tubular protein sits in the center, acts as a scaffold for you to build the whole chromosome. So you take this tubular protein and on this tubular protein, you're going to form loops. Yeah, you're going to form loops with 30 nanometer fibril all around. So it spirals around this chromosome scaffold. Yeah, a 30 nanometer fibril will spiral around the chromosome scaffold. On several such condensation, you get a highly condensed chromosome with a centromere, a short arm and a long arm. So whenever you draw a chromosome with a centromere, a short arm and a long arm, what should you visualize it as? You should visualize it as a central tubular protein or a chromosome scaffold sitting in the center. On this chromosome scaffold, you are forming loops with 30 nanometer fibrils which spirals around this chromosome scaffold. So this is why I was telling you, what did I tell you? I said every chromosome is one long double-stranded DNA which has got condensed with the help of proteins like histones. Is that clear? Yeah. So this is about how chromosomes are formed. Okay, so tell me the answer for this question. The histone that is not present in a nucleosome is. What will be your answer? The histone that is not present in a nucleosome is H1 histone. The next question. 1 second, I'm not able to see the chat. Yeah. So the next one is the most common form of DNA is. Can you answer this question? The most common form of DNA is. What is your answer? Very good, Ankush. Good, Bharat. Yes. Answer is B type of DNA. The most common form of DNA is B type of DNA. So this is about how a DNA can exist in your cell. Okay. So do you know about Watson and Crick model? Yeah, according to Watson and Crick, you know the two strands are exactly complementary and anti-parallel. What do you mean by complementarity? If there is A on one strand, there should be T on the other strand. If there is G on one strand, there should be C on the other strand. So the two strands of any double-stranded DNA will be exactly complementary. What do you mean by anti-parallel? If one strand is 3', 5', the other strand will be 5', 3'. So they are always anti-parallel. They are oriented in a ladder-like fashion. Don't you think that it looks like a ladder? So they are oriented in a ladder-like fashion. The backbone of the ladder, the backbone of the ladder is made up of phosphodiester linkages and ribose or deoxyribose sugars. So the backbone is not only made up of phosphodiester linkages, they are also made up of ribose or deoxyribose sugars. The steps, can you see the steps here? Yeah, the steps are made up of nitrogenous bases linked by hydrogen bonds. So if you are asked what is the linkage which stabilizes a double-stranded DNA, what will be your answer? The linkage which stabilizes a double-stranded DNA is a hydrogen bond. There are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. This is what Watson and Crick stated. Okay, nothing much. Now based on this, there are 7 conformations which are possible. Based on Watson and Crick model, there are 7 conformations which are possible, out of which B type of DNA is the most common physiological form of DNA. Okay, and in B type, for every full turn, what do I mean by full turn? From here till this point, from here till this point. So in B type, for every full turn, there are 10 base pairing. Okay, and every base pairing rises the height by 3.4 angstrom. Okay, every full turn will have how many base pairing? 10 base pairing. Each base pairing rises the height by 3.4 angstrom. So one full turn with 10 base pairing will measure what? It will measure 34 angstrom. And the width will be 20. All of you please memorize all these facts. The width will be 20. And for every full turn, for every full turn, do you see that there is a major groove and a minor groove? Do you see that? For every full turn, there is one deeper groove. The deeper groove is called as major groove. The other one is called as minor groove. So these are the facts that I want you to remember about B type of DNA. So what are the facts I told you about B type of DNA? It's the most common physiological form of DNA. Every full turn will have 10 base pairing. Each base pairing rises the height by 3.4 angstrom. So one full turn measures 34 angstrom and the width will be 20. In B type, for every full turn, there will be a major groove and a minor groove. Okay. The next common conformation that has been asked often after B type, the next common is A type. And you find A type of DNA whenever there is a hybrid between DNA and RNA, or whenever there is a hybrid between RNA and RNA. Do you all understand this? DNA hybrid, double-stranded DNA hybrids will be B type. When will you find A type? Whenever there is a hybrid between DNA and RNA or RNA-RNA hybrid. And in A type, there are 2 classical features which you have to memorize. The first feature is here for every full turn, there are 11 base pairing. In B type, I said there are 10 base pairing. In A type, there are 11 base pairing. The second fact about A type is there is no major or minor groove in A type. In A type, all grooves will be of the same dimension. All grooves are of same dimension in A type. So have you memorized about A type? When did I tell you will find A type? Whenever there is a hybrid between DNA and RNA or RNA-RNA hybrid. Here for every full turn there are 11 base pairing and there are no major or minor grooves here. All grooves are of the same dimension. Now the next type of conformation that can be asked about is Z type. What is the speciality of Z type? It is the only left-handedly coiled conformation. If the question is which is the left-handedly coiled DNA? What will be your answer? It is Z type. Okay, and in Z type, for every full turn there are 12 base pairing. In B type 10 base pairing, in A type 11 base pairing, in Z type 12 base pairing. And you find Z type of conformation only in those regions of chromosome which are rich in GC sequences. Yeah, what is a chromosome? I told you, I said every chromosome is a double-stranded DNA. So this is a chromosome. So if you take a long double-stranded DNA, most of the regions of your chromosome will be equally populated with AT and GC. But very few segments of your chromosome will have more of GC sequences. So those parts of your chromosome which are rich in GC sequences will take up what conformation? They take up Z type of conformation. Okay. So have you understood all about conformations? I see only Bharat answering. Can I see all of you participating in this discussion? Yeah, I always say this, passive learning retention is going to be very low. Yeah, passive learning is you passively listening to a session. Yeah, in which case retention will be very less. So convert your passive learning tendency to active learning. Yeah, active learning is you actively participating in the discussion. So I'll be happy if all of you can type in the chat box. Okay. So that is about all these conformations. So which is the most common physiological form of DNA, all of you? The most common physiological form of DNA is B type. The left-handed helix is seen in, what is your answer? Left-handed helix is seen in Z type. Very good, Ankush. Very good, Ramnathan Bairam. Yes, it is Z type. So now about denaturation of DNA. Yeah, denaturation of DNA is done by all except. So can anybody try answering this? Can you bring about denaturation by increasing the temperature? By increasing, very good Dawood, very good Prithvi. Yes. Increasing the temperature, increasing the salt concentration, decreasing the salt concentration of formamide. Can anybody answer this? Very good Bairam. Good Bharat. Dawood says by decreasing the salt concentration. So I will tell you all that you need to know about denaturation now. But the answer is you can bring about denaturation by any of these except by increasing the salt concentration. Yeah, increasing the salt concentration will only stabilize the DNA. So can you first tell me what is denaturation? Yeah, what is denaturation? Denaturation is a process by which you unwind. Yeah, it's a process by which you unwind a double-stranded DNA to form two single strands. Do you all understand this? What is denaturation? Denaturation is a process by which you unwind a double-stranded DNA to form two single strands. That can be done by breaking what? That can be done by breaking hydrogen bonds. And these hydrogen bonds get broken at higher temperatures. So what is Tm or melting temperature? Tm or melting temperature is the temperature at which 50% of the given DNA can undergo unwinding. Do you all understand this? Tm or melting temperature is the temperature at which 50% of the given DNA can undergo unwinding. And I say this Tm is directly proportional to GC proportion. It is inversely proportional to AC percentage. Will you agree with that? Tm or melting temperature is directly proportional to GC contents, inversely proportional to AT content. Yes or no? It should be yes. Because when there are more number of GC sequences, then there will be more number of hydrogen bonds. So higher will be the temperature that is required to break it. So Tm is directly proportional to GC content, is inversely proportional to AT content. And Tm is also directly proportional to salt concentration because salts generally stabilize any structure. Do you all understand this? Salts generally stabilize any structure because salts can dissociate into oppositely charged ions. Na plus and Cl minus ions. These ions will go and reversely stabilize oppositely charged ions in a structure. So salts generally stabilize any structure. In that case, tell me what will happen if your double-stranded DNA is stabilized with DNA? If your double-stranded DNA is stabilized with DNA, then you will need higher temperature to destabilize it. So Tm is directly proportional to not only GC content, it's also directly proportional to salt concentration. Okay, so not always you bring about denaturation by increasing the temperature. Yeah, only in PCR, only in polymerase chain reaction, we increase the temperature to bring about denaturation. Other than PCR, every other molecular biology technique will be carried out on a gel. On a gel, if you raise the temperature to denature it, the gel will melt. Do you all understand this? Gel will melt. And do you want that to happen? No. So other than in PCR, you don't increase the temperature to bring about denaturation. We add chemicals which can remove the salt. Do you understand this? We add chemicals which can remove the salt, will destabilize the DNA, will cause denaturation. An example for that is formamide. So what is the role of formamide, all of you? Formamide removes salt, it brings about denaturation. So now will you be able to answer this? Yeah, denaturation of DNA is done by all except. Can it be done by increasing the temperature? That is what we do in PCR. Can it be done by decreasing the salt concentration? Yes, that can be done because we have chemicals like formamide to do that. But increasing the salt concentration will not denature it, it stabilizes it. Clear? So the next question. The annealing temperature of PCR is 95 degrees, 60 degrees, 72 degrees and variable. I will wait for 30 seconds for you all to answer. Is it 95 or 60 or 72 or variable? Bharat says variable. Very good, Bharat. Anyone else? Can anyone else try answering this? Yeah, answer is annealing temperature is variable. Yeah, it is not 95. 95 is the temperature. Very good, Dawood. 95 is the temperature for denaturation. 72 is the temperature for elongation. So annealing alone will be variable. So I am going to tell you all that you need to know about PCR. So do you all know that PCR is in vitro replication? Do you know that? PCR is you amplifying a DNA multiple times or you replicating DNA multiple times. But do you know that we have few requirements for replication? All of you tell me yes or no. We have few requirements for replication. What are the requirements? Will you need the polymerase enzyme which replicates? Yes. So you need a DNA polymerase, and your DNA polymerase does not all of a sudden start elongating a DNA strand. It needs a primer to be already present. So what is the second requirement of replication? You want DNA polymerase, you want a primer. Okay. Third requirement is you will need all 4 deoxynucleotide triphosphates. What do you mean by this? Has been repeatedly asked. I have asked you all of the following are requirements of PCR except. Okay. So listen to this carefully. So the third requirement is all 4 deoxynucleotide triphosphates, in the sense you should add deoxy ATP, deoxy CTP, deoxy GTP and TTP. Okay, so why should you add all these? It is because what does your DNA polymerase do? Your DNA polymerase just reads the template strand. And suppose the template strand has got A, G, C, T. What will the DNA polymerase do? It will recruit complementary nucleotides. Tell me what is complementary to A? To A it is T. Yeah, so which form of nucleotide should be attached? It should attach monophosphate nucleotide. But what does it recruit? It recruits complementary nucleotide triphosphates. Why does it recruit complementary nucleotide triphosphates? Because it is supposed to attach 2 adjacent nucleotides. What will be the adjacent nucleotide? Complementary to G is C. So to attach the 2 adjacent nucleotides, what is the linkage that needs to be formed here? The linkage that needs to be formed here is 3' 5' phosphodiester linkage. And for forming this linkage, will you need energy? You will need energy. From where do you get the energy? You recruit complementary triphosphate nucleotides. You break the last 2 linkages. You get energy out of that, and you use that energy to form 3' 5' phosphodiester linkages. So is that clear to all of you here? Yeah, though we know that it's a monophosphate nucleotide that is present in any polynucleotide chain, it is not enough if you supply with monophosphate nucleotides. What should you supply it with? You should supply it with complementary nucleotide triphosphates. Okay, that's the third requirement. And then you need magnesium or manganese to act as a catalyst. The fourth requirement is magnesium or manganese to act as a catalyst. And then you need a buffer to maintain the pH. So can you all tell me what are the requirements of any replication? For any replication, you need the enzyme which replicates, right? You need a primer. You need all 4 deoxynucleotide triphosphates. You need magnesium or manganese to act as a catalyst. You need a buffer. Okay. Now what do we do in PCR? In PCR we simply take a small test tube. Yeah, in PCR what do? We simply take a small test tube. Into that tube we add all these requirements one by one. Okay. And then we place it in an instrument. Okay. So here replication is happening inside the cell or outside the cell, all of you? Here replication is happening inside the cell or outside the cell. It's happening outside the cell. That is why we call PCR as in vitro amplification. All of you tell me what is PCR? It is in vitro amplification. And it is not amplification of a random fragment. It is in vitro amplification of a desired fragment of DNA. So tell me what is the definition of PCR? PCR is in vitro amplification of a desired fragment of DNA. I will give you an example. Okay. Suppose you are involved in molecular diagnosis of Tay-Sachs disease. You are involved in molecular diagnosis of Tay-Sachs disease. A family, okay, a person is coming to you. Yeah, and the person is saying, my family members, many of them are suffering from Tay-Sachs disease. I seem to be normal. I am now 35-year-old. I seem to be normal, but I want to know if I've inherited any defective allele in me, so that I won't transmit. If I get to know this, I will try to take measures to not transmit it to the next generation. Okay, so what are you involved in? You are now involved in molecular diagnosis of which disorder? Tay-Sachs disease. Okay, for this what will you do? You will extract DNA of the person. Okay, when I say I've extracted DNA, I mean to say now you have all the chromosomes of the person, you have all the genes of the person. But for molecular diagnosis of Tay-Sachs, do you want all the chromosomes and all the genes? Come on all of you, for molecular diagnosis of Tay-Sachs, will you need all the chromosomes and all the genes? No, that will be a waste of time and waste of raw material. So instead of meddling, instead of getting confused with all chromosomes and all genes, to get just what is the gene that is mutated in Tay-Sachs disease? The gene that is mutated in Tay-Sachs disease is hexosaminidase A gene. So you want only hexosaminidase A gene. Yeah, let us assume that the black color strand that I have shown you here is the entire chromosome. And in that entire chromosome, just visualize this red color segment. This red color segment as hexosaminidase A gene. So how will you get exactly what you want? By subjecting this to PCR. So in PCR, which is in vitro replication, what will be the first step? For any replication, unwinding will be the first step. And what is the other name for unwinding? The other name for unwinding is denaturation. So in the first step, you are going to unwind this double-stranded DNA to form 2 single strands. This unwinding is otherwise called as denaturation. So the first step of PCR is always denaturation, and we know it has to be carried out at a higher temperature. So the temperature that is used for denaturation is 94 or 95 degree. Is that clear to all of you? The temperature would be 94 or 95 degree. Now after this, your DNA polymerase is going to elongate new strands. And just now I told you, for your DNA polymerase to elongate new strands, the polymerase enzyme needs a primer. So in the next step, you are going to add primers. Okay. And these primers are so chosen. These primers are so chosen that they are complementary only to the flanking sequences of the desired fragment. Yeah, I kept saying that PCR is a specific amplification. So how do we specify the process that this is what I want? You're going to specify the process that this is what you want by constructing primers. How will you construct primers? You choose primers in such a way that they are complementary only to the flanking or the terminal segments of the desired fragment. Okay, can you see that here? Yeah, primers, those blue color fragments are primers. Now you might have a doubt. Okay, it should be complementary to flanking sequences. Why not I choose primers in such a way that they are complementary to the other flanking sequence? This is also a flanking sequence, right? Yeah. So what if you choose primers in such a way that they are complementary to these flanking sequences? That is not good. Yeah, I will give you an example. Suppose AB represents the ends. AB represent the ends of hexosaminidase A gene. Okay, and suppose the top strand is in 3' 5' direction. Yeah, the top strand is in 3' 5' direction. So I have seen to it that my primers are complementary only to 3' flanking sequences. Why should they be complementary only to 3' flanking sequences? Only then the primer sequence will be 5', 3'. Yes or no? Yeah, you know the primer should go on anneal in an anti-parallel direction. So now the primer is in 5', 3' direction. So by mistake, if I choose primers in such a way that they are complementary, very good Bharat. If I choose primers in such a way that they are complementary to 5' flanking sequence, the polarity of primer being 5', 3', and I told you DNA polymerase will synthesize a new strand in which direction? DNA polymerase, any polymerase will synthesize a new strand only in 5', 3' direction. So you are going to get something which you don't want. Do you all understand that? All of you? Is this the same for COVID? I will tell you about COVID in a minute, Dawood. Yeah. So now listen to this. What did I ask you? I asked you, will you be happy getting this side of the fragment? No. Yeah. What do you want? You want only the red color. Yeah, you want only the red color. So how will you choose primers? You choose primers in such a way that, you choose primers in such a way that your primers are complementary only to 3' flanking sequences so that you get exactly what you want. How many primers do we need? You need 2 primers. Okay. And when you add primers, do you see that primers go and anneal? So can we call it as an annealing step? So what is the second step of PCR, all of you? The first step is denaturation. The second step is annealing. Now all of you tell me, how should the temperature of annealing step be? Should the annealing step temperature be high or low? Can you all type it in the chat box? Richapal, Ankush, Dawood, Bharat, anyone? Bairam? Should the annealing temperature be high or low? It should not be high, right? If it's high, the primers will not go and form hydrogen bonds. As when they form hydrogen bonds, it will get broken. But at the same time, the annealing temperature should not be too low as well. What if you said annealing temperature as 5 degrees or 10 degrees? In that case, they will stop, the primers will stop following Watson and Crick base pairing rules. Yeah, you have taken enough caution, to see to it that your primers are complementary only to flanking sequences based on Watson and Crick base pairing rules. So if they don't follow Watson and Crick base pairing rules, your primer will go and anneal somewhere else. And you will get something which you don't want. Yeah, you will get something which you don't want. So to avoid that, it should be optimal. The annealing temperature should be optimal. It should be neither too high nor too low. It should be 5 degrees less than the melting temperature of your primers. Very good, Bharat. It should be 5 degrees less than the melting temperature of your primers. Why? If it's more than melting temperature, primers will melt. If it's too less than melting temperature, there will be non-specific amplification. So annealing temperature should be just less than the melting temperature, 5 degrees less than the melting temperature. For example, if the melting temperature of one primer is 63 degree, the melting temperature of the other primer is 65 degree. Tell me, what is the average? 63 and 65, what is the average? The average is 64. What is 5 degrees less than 64? That will be 59 degree. So for this example which I gave you, the annealing temperature should be 59. But do you think all primers will have the same melting temperature? No. Based on their sequences, their melting temperature would differ. If the melting temperature differs, then annealing temperature will also differ. That is why I said do not blindly follow any of the guides which are available, because I see that many guides say that annealing temperature has to be 55 degrees. It is not always 55 degree. Do you all understand that? It is not always 55 degree. It's just 5 degrees less than the melting temperature of your primers. It is variable. Now, after primers have annealed, your DNA polymerase is going to elongate new strands. So, what is the third step of PCR? The third step of PCR is elongation, because in this step your DNA polymerase will elongate new strands. And for this you will have to use a thermostable DNA polymerase. Yes or no? Why should you use the thermostable DNA polymerase? Because the DNA polymerase should be capable of withstanding cyclical rise of temperature to 94 or 95 degree. How we know melting temperature? That's a very good question, Jayalakshmi. So it is you who will be ordering for your primers, yeah, depending upon which gene you are working on. For example, here I was saying hexosaminidase A gene is what we are working on. So for hexosaminidase A gene, there will be flanking sequences. So you will make primers, you will specify this is the primer sequence I want. So you know its sequence, right? Once you know the sequence, don't you know that TM is directly proportional to GC content and inversely proportional to AT content? It is directly proportional to salt concentration. Jayalakshmi? Yeah, so based on the facts, what is the percentage of GC? What is the percentage of AT? What is the percentage of salt? Yeah, you have a formula to calculate melting temperature. Or actually, whenever you ask for primers, when a manufacturer manufactures and gives you primers, that vial, yeah, that vial will have an information. It will tell you the melting temperature of the primer which I have manufactured is this. Do you get it, Jayalakshmi? And how do they find out is doing experiments and based on this. Is that clear? Okay, so that's about melting temperature. Now about elongation, yes, it has to be thermostable because it has to withstand cyclical rise of temperature to 94 or 95 degree. And the human DNA polymerase is not thermostable. So we have extracted DNA polymerase from an organism which lives in hot springs. And that organism is Thermus aquaticus. Yeah, that organism is, my pleasure, Jayalakshmi. That organism is Thermus aquaticus and hence the enzyme is Taq DNA polymerase. Okay, and Taq DNA polymerase, optimum temperature required for Taq DNA polymerase to elongate new strand is 72 degree. Yeah, and that is why elongation step is always carried out at 72 degree. So have you understood all these temperatures? Yeah. So denaturation, what is the temperature, all of you? For denaturation it is 95. For annealing, it is 5 degrees less than the melting temperature of your primers. And then for elongation, it is 72 degree. Okay. And the instrument that you use for PCR is called as thermocycler. Why do you call the instrument as thermocycler? Because the instrument cycles between temperatures. It does not do any other magic. All that the instrument does is first it raises the temperature to 95, reduces it to annealing temperature, increases it to 72. Again increases it to 95, reduces it to annealing 72. So it cycles between temperatures. That's why the equipment is called as thermocycler. Okay. And someone was asking about SARS-CoV-2 diagnosis, right? So what do we do for SARS? I missed the name. So for SARS-CoV-2 diagnosis, yeah, I saw it. One second. Dawood. So Dawood had this doubt. So for SARS-CoV-2 diagnosis, it is reverse transcriptase qPCR. Yeah, what is that we do for SARS-CoV-2 diagnosis? It is reverse transcriptase qPCR. So what does it mean? Do you all know that your coronavirus, yeah, it is an RNA virus, okay? And handling RNAs is difficult. Amplifying RNA is difficult. So what you do first is you convert this RNA to DNA. You convert the single-stranded RNA of SARS-CoV-2 virus to double-stranded DNA. That is done by reverse transcriptase enzyme. And after you convert it into double-stranded DNA, then you subject it to qPCR. What is qPCR? It is quantitative real-time PCR. Do you all understand this? qPCR is quantitative real-time PCR, which does not worry about amplifying. It actually worries about quantifying the nucleic acid. Okay, and for quantifying the nucleic acid, we use Stackman probe. What is that we use? We use Stackman probe. Okay, so this Stackman probe will have a fluorescent dye. So as and when amplification happens, the fluorescence keep increasing. Do you all understand this? As and when amplification happens, the fluorescence that is red will keep increasing. And we find out something called a CT value. Have you all heard of it? Yeah, what is CT value? CT value is cycle threshold. So what is cycle threshold? We always have a minimum fluorescence intensity for a PCR. Yeah, we always have a minimum or a bare minimal fluorescence threshold fluorescence intensity. So what is CT? CT is the cycle. It is a cycle at which it crosses the minimum fluorescence intensity. Do you all understand this? What is CT? CT or cycle threshold is a cycle at which the fluorescence intensity crosses the threshold fluorescence intensity. If the cycle is less, suppose I say CT value is 15, what do I mean? I mean to say even in the 15th cycle, even in the 15th cycle it has crossed the threshold intensity, which means what do you think about the dose? What do you think about the viral load? The viral load is more. Yeah, suppose I say CT value of this sample is 35. What does that mean? It means only at the 35th sample. No, only at the 35th cycle. Yeah, only at the 35th cycle the fluorescence intensity crosses the threshold intensity. So it takes such a long time for it to reach the threshold intensity, which means what do you think about the viral load? The viral load is less. Do you all understand this? So that is CT. So many of you have been asking me for CT. What is CT? CT is cycle threshold. It is a cycle at which the fluorescence intensity crosses the threshold intensity. If that cycle value is low, it means even at such low number of cycles, from first cycle, second cycle, third cycle, the 10th cycle it crosses, which means the viral load is high. If the CT value is high, viral load is low. Is that clear? So that's called as reverse transcriptase qPCR. Okay, so we use Stackman probe. So what is answer? The annealing temperature of PCR is variable. All the following are, okay, I have another session. So all the following are requirements of PCR except. Can anybody give me the answer? Because we have discussed in detail about PCR already. I'll be happy if any of you can give me the answer. Very good, Dr. Happiness. Oh, is that the answer for the previous question? All of the following are requirements of PCR except. Good Bharat, good Prithvi, good Dawood, yes. Yes, what is answer? It is restriction enzyme. So what are the requirements of PCR? I already told you. Requirements of PCR will be DNA polymerase enzyme, primers, all 4 deoxynucleotide triphosphates, magnesium or manganese as a catalyst and a buffer. Okay, you are not going to use restriction enzyme. When will you use restriction enzyme? You use restriction enzyme only for RFLP. Yeah, you use restriction enzyme only for RFLP or for recombinant DNA technology, not for PCR. Okay. So this is an interesting question and also a repeatedly asked question. siRNA causes what? Gene knockdown, gene knock-in, gene knockout, gene conversion. What is your answer? siRNA or small interference RNA, they mediate interference phenomenon. So this interference phenomenon will result in what? Very good, Bharat. Dr. Happiness says it's gene knockout. Ankush says it's knockdown. Yeah, the answer is gene knockdown. Yeah, siRNA causes what? Gene knockdown. So let me explain this to you. What is siRNA? siRNA interferes. Interferes with what? It interferes with gene expression. Do you all understand this? siRNA mediates interference. Interference is nothing but interfering with gene expression. So what is gene expression? Gene, listen to this carefully because I am going to tell you about what CRISPR does now. Okay. So what is gene expression? Gene giving rise to mRNA on transcription and mRNA on translation forming proteins. Yeah, transcription followed by translation forming proteins. This is gene expression. So whenever you say something interferes with gene expression, you should specify at what level it interferes. And your siRNA interferes with gene expression at translation level. Do you all understand this? It interferes with gene expression at translation level. So how does it do that? Listen to this. These siRNAs, like any other RNA, will have its own specific sequences. Do you all understand this? These siRNAs, like any other RNA, will have their own specific sequences. And suppose these siRNAs find complementary sequences on any of the fully formed mRNAs. If they find complementary sequences on any of the fully formed mRNAs, these siRNAs go and hybridize with those mRNAs. Not knowing this, your ribosome, yeah, you know ribosome reads the mRNA, yes? Yes or no? All of you, can you all say yes or no? What do ribosomes do? Ribosomes read the mRNA from 5' end to 3' end. They try to translate these mRNA. So as they are proceeding on the mRNA, they come across this double-stranded RNA structure in between. And ribosome knows that finding a double-stranded RNA structure on the way during translation is not normal. Do you understand this? Ribosome knows that finding a double-stranded RNA structure on the way during translation is not normal. So this ribosome will recruit RNA transcription silencing complex. What will it recruit? It will recruit RNA transcription silencing complex. And as the name indicates, one of the complex, one of the proteins of this complex is an endonuclease. The silencing complex, one of the proteins is an endonuclease. That endonuclease makes multiple nicks on the mRNA. So this mRNA is fragmented. Once mRNA is fragmented, will it be translated, all of you? Once mRNA is fragmented, will it be translated? No. So what have you interfered with? You have interfered with, come on, gene expression. At what level? At translation level. Is that clear to all of you? Yeah. So what do siRNAs do? Can you all answer this? siRNAs interfere with gene expression. At what level? At translation level. So how do they interfere with gene expression at translation level? I said these siRNAs have their own specific sequences, and if they find complementary sequences on any of the fully formed mRNAs, they hybridize with those mRNAs by forming hydrogen bonds. Not knowing this structure, ribosome happily translates this mRNA and it proceeds on the mRNA until it comes across this double-stranded RNA structure. If it finds double-stranded RNA structure, quickly ribosome recruits what? It recruits RNA transcription silencing complex. One of the proteins is an endonuclease. So that endonuclease fragments the mRNA, does not allow the mRNA to be translated. So do you think this will result in a disease? When do you think this should happen? If this happens, will it result in a disease? No. Okay. It is a form of regulation. It's a form of regulation of gene expression. siRNA is a form of regulation of gene expression. I will give you an example. Okay. Suppose there is an inflammatory focus in a person. Listen to this. Suppose there is an inflammatory focus in a person. And this inflammatory focus has switched on multiple cytokine genes. The inflammatory focus is switched on multiple cytokine genes. And these genes have given rise to cytokine mRNAs. Transcription is over, cytokine mRNAs have been synthesized. Okay. Suddenly something has happened or somebody has intervened. And what is removed? This inflammatory focus has been removed. Yeah, once the inflammatory focus is removed, will you want the mRNAs to be translated as proteins? Once the inflammatory focus is removed, will you want the cytokine mRNAs to be translated as proteins? No. If you allow that to happen, that is going to result in unwanted cytokine storm. Yes or no? It is going to result in unwanted cytokine storm. So transcription is already over, but you don't want translation to happen. Right? Transcription is already over. You don't want translation to happen. What will you do? You will immediately switch on interference phenomenon. So all these functional genes, for example, cytokine genes, they have in the intronic sequences, they have their corresponding siRNA genes. So only till some time back we thought introns are non-coding. Haven't we been taught that way? Yeah, we have always been taught that introns are non-coding intervening sequences, right? But now we are trying to pick up, we are trying to find out more and more functions for introns. One of the functions identified is these introns will have sequences for the siRNA genes. Okay, so those introns will be transcribed and what will be produced? siRNAs will be produced. And what will these siRNAs do? Once they are transcribed, these siRNAs will go and hybridize with those mRNAs. And mRNAs will not be translated as proteins. This is called as interference. So in interference, are you altering the gene sequence? All of you, in interference, are you altering the gene sequence? Can anybody tell me? We are not altering the gene sequence. Only when you alter the gene sequence, you can call it as gene knockout or gene knock-in. Here we are not touching the gene sequence at all. What we are doing is we are down regulating gene expression alone. Do you understand this? What we are doing here is we are only down regulating the gene expression. So what will you call it as? You have to call it as knockdown. When will you call it as knock out or knock in? Knock out or knock in when you alter the gene sequence. That's not being done here. So have you understood this? siRNAs cause what? siRNAs cause gene knockdown. Okay. So now about, yes, at the translation level. Very good. So now about CRISPR mediated gene editing, because we are discussing about knockdown. Let me tell you about knockout and knock in. So do you all know that CRISPR, it has an endonuclease. Yeah, CRISPR-Cas enzyme system. Cas is an endonuclease. So what does this endonuclease do? It will cause a double-stranded DNA break. Okay. So whenever you are trying to edit a gene, do you all know the function of CRISPR-Cas enzyme system? Do you all know the CRISPR-Cas enzyme system? If you don't know about the application of CRISPR-Cas enzyme system, using an endonuclease which is available within the system, we try to edit gene. Do you understand this? We try to edit gene. How do we edit gene? This endonuclease makes a nick at a specific site. Okay, so what does it cause? It causes double-stranded DNA break. And whenever there is a double-stranded DNA break, you must have learnt in repair mechanism that it can be done by, the repair can be done by either non-homologous end joining, yeah, or it can be done by homologous DNA repair. So tell me, what are the two ways by which any double-stranded DNA break can be repaired? It can be done by either non-homologous end joining or by homologous DNA repair. And depending upon whether we do it by non-homologous end joining or homologous DNA repair, your CRISPR gene editing mechanism can either result in gene knockout or gene knock-in. Okay, what do you mean by gene knockout? Can anybody tell me what is gene knockout? In this case, the defective gene is removed. Isn't that self-explanatory? In gene knockout, what do we do? In gene knockout, we just remove the defective gene. Whereas what is gene knock-in? The defective gene is replaced with the normal gene. In gene knock-in, we replace the defective gene with the normal gene. Okay. So when will it result in knockout and when will it result in knock-in? If it is non-homologous end joining, very good. If it is non-homologous end joining, there will be always deletion of few gene sequences. Okay, non-homologous end joining, when you try to link two non-homologous ends, You will remove few sequences and then you connect them. So there is going to be loss of few gene sequences. So what will it result in? It is going to result in knockout. Do you all understand this? Non-homologous end joining results in knockout. Whereas if it is homologous DNA repair, yeah, have a look at this. Suppose this is where there has been a defect. This is where there has been a defect. You have used CRISPR-Cas enzyme system, and this CRISPR-Cas enzyme system, what it has done is it has deleted it. I'm sorry. No, this is not what I wanted. Yeah, suppose the CRISPR-Cas enzyme system has cut it, has done a double-stranded DNA break. What do you mean by homologous DNA repair? In homologous DNA repair, the repair mechanism is specified. The repair mechanism is specified by you with a homologous normal sequence. So when you provide homologous normal sequence, your DNA polymerase will use this as a template and will exactly substitute the normal sequence here. Yeah, what will it do? It will exactly substitute the normal sequence. So here you have not only removed the defective gene, you have also substituted a normal sequence. So what will you call it as? You will call it as knock-in. Okay, homologous DNA repair will result in gene knock-in. Non-homologous end joining will result in knockout. So the summary here is, I am going to summarize it. What does interference do? All of you, can you tell me what does interference do? Interference is down regulating gene expression. So interference is gene knockdown. Okay. Non-homologous end joining, you are going to delete few gene sequences. So non-homologous end joining results in gene knockout. Homologous DNA repair will result in gene knock-in. Is that clear? Yeah. So these are the MCQs that can be asked you as far as interference and CRISPR mechanisms are concerned. So I will discuss this. Let this be the last question for today's session. Yeah. So mitochondrial DNA is, what is your answer? Can anybody give me a right answer here? Mitochondrial DNA is, no answers? It is a closed circular DNA. Yeah, mitochondrial DNA is closed circular. Very good, Prithvi. Yes. So let me tell you few facts about mitochondrial DNA which have been asked previously. It is not curvilinear. Yes, it is closed circular. Okay. So what are the facts that you need to know about mitochondrial DNA? As I just now told you, it's a closed circular DNA similar to plasmids of prokaryotes. Okay. So we actually consider mitochondria. We actually consider mitochondria as a prokaryote which has gained entry into eukaryote. But because this mitochondria provides us with some survival advantage, we have allowed it to get into symbiosis. Do you all understand this? So mitochondrial DNA is a closed circular DNA similar to plasmids of prokaryotes. And though mitochondria has got its own DNA, not all proteins of mitochondria are coded by the same. Your mitochondrial DNA codes only for 13 proteins of electron transport chain. They code only for 22 tRNAs. They code only for 2 rRNAs. Yeah, what do they code for? They code for 13 proteins of electron transport chain. They code for 22 tRNAs. They code for 2 ribosomal RNAs. Okay. And then the genetic code here is quite different. Do you know that? Yeah, mitochondria. I mean, why should you know that they code only for this? Because only then you will understand. Yeah, only then you will understand that your mitochondria is not self-sufficient. For most of the proteins, for most of the RNAs, your mitochondria is dependent on nuclear DNA. So if you take out mitochondria, leave it as such, it cannot replicate because it's not self-sufficient. Okay. And the genetic code here is quite different. What is UGA, all of you? Usually UGA is a stop codon. Okay. In mitochondrial DNA, it codes for tryptophan. UGA codes for tryptophan. It's not a stop codon. Usually AGA and AGG code for arginine. So what is the difference in genetic code? I've told you the first difference is UGA is not a stop codon but it codes for tryptophan. Okay, AGA and AGG usually code for arginine, but both act as stop codons here. Okay, so genetic code is different. And mitochondrial DNA is more prone to get mutated. Reason is DNA polymerase gamma. This can be asked to you. They can ask you which DNA polymerase synthesizes mitochondrial DNA. What should be your answer? DNA polymerase gamma. It does not have proofreading and repair activity. Yeah, it does not have proofreading and repair activity. That is why mitochondrial DNA is more prone to get mutated. And such mutations are only maternally inherited. Very good, Dawood. It is only maternally inherited. It is because during zygote formation, all that the sperm contributes with is, yeah, the sperm contributes with only its pronucleus. So the remaining parts of sperm will be shed down. So even if the sperm's mitochondrial DNA has been mutated, because it does not get into zygote formation, it is not going to be inherited. Do you understand this? So mitochondrial DNA mutations are only maternally inherited. So these are the facts that you need to know about mitochondrial DNA. So what are the facts I told you? I said it's closed circular. It codes only for 13 proteins, 22 tRNAs and 2 rRNAs. The remaining proteins and RNAs are coded only by nuclear DNA. Genetic code here is different. UGA does not code for, does not act as stop codon, but it acts as tryptophan. AG and AGG do not code for arginine, but they act as stop codons. They are more prone to get mutated because DNA polymerase gamma does not have proofreading and repair activity, and such mutations are only maternally inherited. Okay. So all the following are properties of genetic code. Can we discuss this or do we break? All of you? I leave it to you. All the following are properties of genetic code except degeneracy, ambiguous, not punctuated, non-overlapping. What is the right answer? Can anybody pick a right answer here? We know for sure our genetic code is degenerate. Yeah, it is unambiguous. Our genetic code is unambiguous. So this is a false statement. Yes, it is not punctuated. It is non-overlapping. Okay. So let's try to understand properties of genetic code. The first property is degeneracy. So can you tell me what is degeneracy? Okay, let's discuss. Yeah, I've started. Yeah, what do you mean by degeneracy? Can you first tell me how many choices of nucleotides do we have to make a codon? Totally we have 4 choices of nucleotides, right? A, U, G, C. Totally we have A, U, G, C. And out of these 4 choices, you make a combination of 3 to form a codon, right? You make a combination of 3 to form a codon. So how many such combinations are possible? We have 4C3 combinations. What do you mean by 4C3 combinations? 4 to the power of 3. What is 4 to the power of 3? It is 64. So totally how many codons do we have? Yeah, totally we have 64 codons. And out of 64 codons, how many are stop codons? Amber codons or stop codons? There are 3 stop codons. So how many codons code for amino acids? The remaining 61 codons code for amino acids. How many amino acids do we have? 20 amino acids. So 61 codons code for just 20 amino acids. Do you all understand that here? Yeah, 61 codons code for just 20 amino acids, which means more than one codon can code for a single amino acid. Is that clear? Yeah, more than one codon can code for a single amino acid. That is called as degeneracy. Now, how can more than one codon code for a single amino acid is being explained by wobble phenomenon. Yeah, degeneracy is explained by what? It is explained by wobble phenomenon. So, what is wobble phenomenon? Do you remember I told you about ionosin when we started the session? Yeah, so this is where ionosin comes into play. So what does wobble phenomenon state? According to wobble phenomenon, the third nucleotide of anticodon of tRNA, just listen to this even if you don't understand it because I'm going to show you how in a minute. Okay. For now, what is wobble phenomenon? According to wobble phenomenon, the third nucleotide of anticodon of tRNA is most commonly inosine monophosphate. And this inosine monophosphate does not follow Watson and Crick base pairing rules. Don't we know that already? Yeah. The third nucleotide of anticodon of tRNA is most commonly inosine monophosphate, and this does not follow Watson and Crick base pairing rules is something which we know already. Now, how can this simple fact, how can this simple fact explain degeneracy? And for that, let's have a hypothetical mRNA. Have a look at this. We have a hypothetical mRNA with just 4 codons. And for your information, all these 4 codons code for the same amino acid glycine. All these 4 codons code for the same amino acid glycine, and that's what we are trying to explain how. It is because the tRNA, listen to this carefully, the tRNA which carries glycine along with it. The tRNA which carries glycine along with it comes with an anticodon CCI. So all of you tell me, what is the anticodon for glycine? The anticodon for glycine is CCI. So do you see that inosine monophosphate is the third nucleotide of anticodon of this tRNA? And don't you know that this does not follow Watson and Crick base pairing rules? So this tRNA will be least bothered. This tRNA will be least bothered about what is present as the third nucleotide in all these 4 codons. The tRNA will just check if it can follow Watson-Crick base pairing rules with respect to first and the second nucleotides. With respect to CC, do you find GG everywhere? With respect to CC, do you find GG everywhere? Yes. So this tRNA will happily go and hybridize with all these codons. And it will attach what as the amino acid? It is going to attach glycine as the amino acid sequentially. So we are going to get a polyglycine chain. Do you all understand this? We are going to get a polyglycine chain. So now do you understand how can more than one codon code for a single amino acid, all of you? Now do you understand how can more than one codon code for a single amino acid? This is how it is. Okay. So can you tell me what is degeneracy? I will explain this in detail again. What is degeneracy? Degeneracy is more than one codon coding for a single amino acid. How can more than one codon code for a single amino acid is explained by which phenomenon? Wobble phenomenon. And according to wobble phenomenon, I said the third nucleotide of anticodon of tRNA is inosine monophosphate, right? But that's only for you to understand. Can you tell me what is the polarity in which mRNA takes part in translation? What is the polarity in which mRNA takes part in translation? 5', 3' polarity. So what will be the polarity of your tRNA? Can you all answer this? What will be the polarity of tRNA? It is 3', 5'. Okay, so when you write the sequences of anticodon and 3', 5' polarity, your inosine monophosphate might look as if it's the third nucleotide. Okay, but in genetics you are supposed to write any polynucleotide chain sequence only in 5', 3' polarity. You are supposed to write sequences of any polynucleotide chain only in 5', 3' polarity. It can be template strand sequence, it can be coding strand, mRNA, tRNA, rRNA, whatever it is, you are supposed to write them only in 5', 3' direction. So if you start writing it in 5', 3' direction, do you see that your inosine monophosphate is actually the first nucleotide? Yes or no? Inosine monophosphate is actually the first nucleotide of anticodon of tRNA. And that is the basis of wobble phenomenon which explains degeneracy. So how will you remember it? Inosine is like Roman letter 1, right? I is like Roman letter 1. So inosine monophosphate is the first nucleotide of anticodon of tRNA that does not follow Watson and Crick base pairing rules. Okay. So now I will tell you wobble phenomenon in 3 statements. I will tell you 3 statements for wobble phenomenon. I want you to tell me whether those 3 statements are right or wrong. Okay. The first statement is according to wobble phenomenon, the third nucleotide pair formation is not specific. Yes or no? According to wobble phenomenon, there is a reduced stringency. Yeah, there is a reduced stringency when the third nucleotide pair is formed between codon and anticodon. Is that true or false? There is reduced stringency when the third nucleotide pair is formed between codon and anticodon. Yes or no? Yeah, it's a true statement. The second statement is, according to wobble phenomenon, the third nucleotide of codon is not specific. Yeah, according to wobble phenomenon, the third nucleotide of codon of mRNA is not specific. That is true. It's also true. Third is, according to wobble phenomenon, the first nucleotide of anticodon of tRNA is not specific. That is also true. Yeah, this is why you often get confused about wobble phenomenon if you try reading it from a textbook. So multiple textbooks give you multiple versions of wobble phenomenon. They are all right, but we get confused. Okay. So tell me, what is wobble phenomenon? There is reduced stringency when the third nucleotide pair is formed between codon and anticodon. According to wobble phenomenon, the third nucleotide of codon is not specific. The first nucleotide of anticodon is not specific. Okay, good. So this is degeneracy. That's one property. Second property of genetic code is it is unambiguous. What do you mean by unambiguous genetic code? Though we say more than one codon can code for a single amino acid, can the reverse be true? Yeah, will you ever say one codon can code for more than one amino acid? Yeah, will you ever say one codon can code for more than one amino acid? In that case, you don't need any code at all. Right? So your genetic code is degenerate, but it is unambiguous. And the third property is it is non-overlapping. What do you mean by non-overlapping genetic code? Will you ever say I will use first, second and third nucleotide as the first codon, and I am going to use third, fourth and fifth as my second codon. Let me use 4, 5 and 6 as my third codon. Do you ever overlap one codon with another? We don't do that, right? That is why we say it is a non-overlapping genetic code. And then it is not punctuated. What do you mean by not punctuated? After every 3 nucleotides, do you have a punctuation like this? Yeah, after every 3 nucleotides, do you have a punctuation like this? No. Had it been punctuated like this, what would have happened? Had it been punctuated like this, and if there is a loss of one nucleotide anywhere here, then the amino acid coded by this codon alone should have been defective. Right? Whereas all the other codons should have coded for normal amino acids. But is that what happens? No. What happens? Whenever there is a loss of one nucleotide anywhere, the entire frame of reading will get shifted from that place. Don't we know that? The entire frame of reading will get shifted from that place. So just because frame shift mutation is possible, we know our genetic code is not punctuated. Okay? And it is universal. The last property is it is universal with few exceptions. What do you mean by universal genetic code? Will you ever say, okay, AUG can code for methionine only in liver, but I am going to use AUG for tryptophan in some other cell. Will you do that? No, right? AUG codes for methionine in all your cells. That is what we mean by our genetic code is universal across all cells, all tissues. But with few exceptions. What is the exception I told you? The exception is mitochondrial DNA. What did I tell you about mitochondrial DNA? In mitochondrial DNA, UGA is not a stop codon, but it codes for tryptophan. AGA and AGG act as stop codons. So it is generally universal, but with few exceptions. Okay. So tell me, what are the properties of genetic code? Very good, Bharat. You are right. So what are the properties of genetic code? Your genetic code is degenerate, but it is unambiguous. It is non-overlapping. It is not punctuated. It is universal with few exceptions. Okay. There are 2 exceptions for degeneracy. Though I said your genetic code is degenerate, meaning more than one codon can code for a single amino acid. Exception is methionine and tryptophan. For methionine and tryptophan, there is only one codon each. Okay, for methionine tryptophan, there is only one codon each. So, which is what are the amino acids which do not follow degeneracy? Methionine and tryptophan. Okay. So, what is answer? All the following are properties of genetic code except ambiguous. It is not ambiguous, it is unambiguous. Okay. So, with this, let me close the session for today. Yeah. So, I hope you have a clear idea or at least an overview of genetics now. Yeah. So, with this overview, you can try solving as many MCQs as possible. Okay. If you have any doubts, you can ask me through either Unacademy community or through my Telegram group. Okay. Thank you all.